And we're very grateful to Sabrina Leslie for joining us from Vancouver, I think, where it is 6:30 in the morning. So please be considerate with your questions. Okay, so over to you, Sabrina.
Okay, thank you very much for the invitation, and I'm glad to be here before the sun rises in Vancouver. Let me see, so I'm gonna share my screen. Okay, great. Thanks. Yeah, so as was mentioned, I'm in Vancouver. We actually just moved during the pandemic. We're at the Michael Smith Laboratory, which is a biotechnology laboratory, and the physics department. And as was introduced, we're really at the microscopic scale. So we're excited to be here. I think the pandemic has especially shown the world how Vancouver is a center of development of lipid nanoparticles, which have played a key role. And I was going to share today some of our work in that area, which should be in press soon. So the big picture of my research group is to establish a through line of understanding. So from this most microscopic level of detail, looking at individual molecular interactions, to sort of a more mesoscopic level of detail of imaging molecules being delivered in their carriers inside lipid particles into cells and eventually targeting into tissues. And our move to a more biological environment around us gives us the infrastructure to do all these things. For those who are familiar with our work or have seen me speak before, most of my presentations have been zooming in on molecules binding to molecules, which we image directly in our way. And in this talk, I was going to introduce some of our new work on imaging lipid nanoparticles carrying those molecules. So in my group, we have microscopes dedicated to looking at, for example, oligo probes or antisense oligo binding to RNA targets in little wells. We're also looking at lipid particles containing those drugs with those drugs loaded. And finally, we're developing infrastructure to connect that microscopic detail to the level of entry into tissue. And that's very exciting for us. That's a new phase. So as I mentioned, we have this 3 axes of work. And in the middle of this slide, you see a movie of an antisense oligo and RNA. So we literally look at them binding and unbinding as a function of modifications to the drug or the target, asking questions about biophysics of interactions. And then at the bottom, you see a little movie of a lipid particle, which is containing some RNA drugs. And at the top, you get the sense that if we use technology we've developed, we can look at large arrays of these interactions and count complexes over time. But today, what I'd like to do is to focus in on our work on, in great detail, checking out these lipid particles containing RNA drugs, which has become a detailed biophysical study, the outcome of which should be very exciting applications in this vaccine and genetic medicine area. And this work isn't in press yet, but we hope it will be soon. So this is a video of how we isolate and look at many individual particles or molecules. What you're seeing is the species are floated in a little flow cell, and the flow cell is composed of 2 sheets of glass. The top one we deflect actually with a nano positioner with an inverted lens. So it's called convex lens induced confinement. So there's a curved surface above and a flat surface below. You bend the glass together, and inside any surface of the glass, and more conveniently the bottom, because we're not bending it, we can embed features like wells. So what you're seeing here is fluorescently labeled nanoparticles, first loaded in a flow cell of 2 sheets of glass, and the roof is bending down, and then they're entropically trapped. So even if you leave a thin layer of liquid, they won't escape the wells. To them, the wells are very big, and that thin layer might be so small that they're sterically blocked from escaping. So we can watch them for a long time, can track them, look at things bind to them, look at how much drug's loaded on them, do reactions in real time. And this technique we call CLiC. And yeah, so here's a little schematic of the technique just to orient you to other data in this talk. So there's a nano positioner with a little lens on the bottom. It squeezes the top sheet of glass down, and we're looking up into this flow cell. So in other work that we've published, we've confined polymers and long lines. It's a neat technique for single molecule resolution in genomics without tethers or fields. We trap molecules to look at their interactions, and today we're trapping nanoparticles. And this is the instrument that's mounted on our microscopes in the lab for performing CLiC. There's been various interests from other groups as we've developed our research, and especially in Canada. Sorry, this slide is has some annotations from another meeting. People are pretty interested in trying it out themselves. So with some of my trainees, we spun out a company to provide it. It's now in 10 other labs looking at other things, but it's the same one we use for our research. Yeah, and I guess for many people at this meeting, we have an understanding, but yeah, what's really often missing in therapeutics work or any biochemistry work and can help for the optimization of important molecules or carriers is this single molecule information. So whereas you might make one measurement of a rate in SPR or bulk fluorimetry, or see an average result over multiple populations, when we zoom in and count the complexes, maybe there's folded molecules that aren't binding as well and unfolded molecules that are, we can see the distribution of states. We can follow events in time. If there are multiple kinds of reactions going on, we can identify them and really get all this detail. So that's the motivation for the work. And then in moving to Vancouver and collaborating with Peter Cullis and Precision Nanosystems and other people at the center of these lipid particle development, we've gotten to learn how we can help with those as well. So there's a particular interest in delivery, the lipid particle size, their dynamics, just being able to watch them fuse to artificial endosomes, release in the endosome, and ultimately cells is territory that we can really help with, and then connects with the end result of the molecular interaction. So that's what I'm going to focus on in this talk, developing a platform for that and first biophysical studies of these lipid particles carrying these RNA drugs. So I'm going to now move to those results, and they should be hopefully in press soon. Yeah, so here, similarly to before, we are using the CLiC platform to load and squeeze the nanoparticles into wells for observation. We design the wells so that we can track a full trajectory for a long time. So they're typically a few microns wide here, and the depth is around the focal depth. So that's a few hundred nanometers typically is convenient. And the particles are just diffusing in these wells. And from the diffusion trajectory, we can track their diffusion coefficient and extract the size based on the well geometry. And from their brightness, if the RNA or other drug is loaded, we can extract the loading. So that gives us a lot of information. We can also continually replenish the wells. So we get very high statistics. And we found these populations, molecules, particles, biological material, can be messy and heterogeneous. So this required us to develop a simulation platform to interpret our data, and of course, validate it on small particles, which were well known. So what we did for that is we used small beads, 50 nanometer beads, and we can actually create an artificial movie of a diffraction limited spot. So the bead diffuses around as a diffraction limited spot. And we can simulate kind of the noise level in our images that we choose for a particular experiment, and understand what data would come out if it were a perfect bead of a certain size. And then we can compare that to the real data and be very confident that our interpretation of our data is giving us result that we would get if it really was a bead of that size, if that makes sense. So that was very important. And it highlighted some of the confinement physics that we have to understand to simulate these things. And while that is related to our technology, it is also the case that in cells and in these biological environments, confinement physics is really important too. So this is giving us a lens in studying particles and their motion in very cell-like environments, which is ultimately our goal. So from a long trajectory, as a particle searches this well, we calculate the mean squared displacement curve. So at short times, it's a straight line. And in free space, you'd be familiar with mean squared displacement equals 4 times the diffusion coefficient times time in free space. But in our well, obviously, the area is finite. So there's a theory that we developed, and our geometry is a little special to extract that curve. And then we had to really understand, is there a confinement effect of our wells in these results? From a practical point of view, if we're going to then analyze many kinds of lipid particles containing drugs, we'd love to operate in a regime where the confinement effects were minimal, but we were pulling out results. And that regime, turns out, does agree so the diffusion coefficient, the native diffusion coefficient, agrees with the measured one for the larger wells that we've been using. So what you're seeing here is data that we've collected for wells of different depths. So 200, 350, 500, 1200 nanometers deep, and there's several microns width, which is very wide. And we see that the distribution of diffusion coefficients is fairly similar for the larger depths, but is altered for the lower depths. And that's based on hydrodynamic interactions with the surface. But for these larger depths and the wells that we practically use, the altered diffusion coefficient because of confinement has a very small effect. So this study took some work, but in the end, from the practical standpoint, we can operate with low confinement effect in terms of pulling out the particle size. And this is just looking a little bit more detail. So when you're looking at diffusion and loading and dynamics in confined conditions, you have to make sure you understand the effect of that confinement. But in our device, for a few micron wide well and several hundred nanometers deep for these roughly 50 to 80 nanometers lipid particles, you have little effect. And this data here is shown with 50 nanometer beads. So that's something we learned. And then in terms of working with the lipid particles containing the drugs, we have the ability to count them with precision to single drug per particle by using photo-bleaching. So what's shown here is now the lipid particle, so size on the order of 80 nanometers. The recipe is the Onpattro, or very similar to Madonna or Pfizer recipe. It contains siRNA drugs. So the figure A here on the left has 1, and we've turned on a laser and bleached 1 step. This one has a few, this one has 10. So for every particle, as we image a long track, we can see these photo bleaching steps and count out the number of drugs in the particle based on photo bleaching. And then we can construct essentially the distribution of drugs for this particles. And for some of our next steps are to look at mRNA loading, which are these macromolecular vaccines or drugs, and also like self-amplifying RNA and other very large polymers. And detailed studies of that loading can be very much refined with this technique and are often missing. And this is something we can do. There is some variability in the single step of the photo bleaching, but we create a distribution and then choose one number to normalize by. Okay, so now moving to the studies that we've done. Essentially, we wanted to look at some example lipid particles. I apologize, this Zoom screen I think is blocking all my titles. It is 6:30 in the morning, sorry about that. Oops, the way I've displayed this, that is, I don't know how to get rid of that. Okay, so for our first results, we looked at the lipid particles with the Onpattro recipe, and they're loaded with a dye, which is an analogous dye to Cy3, Cy5, It's used in other standard work and published work by the Peter Cullis group. So this dye is lipophilic, so it's in the membrane of the lipid particle or the outer layer. And if you look at the figure on the right, in this particular circumstance at this pH buffer, so we're looking at two different pHs, we expected the dye to be really in the outer layer of the lipid particle. And in that case, if we look particle by particle, and this was a test for our sensitivity, we should see that the intensity scales as the radius squared of the particle. So for every particle, we can pull out its diameter or radius from its long diffusive track, and then we can count or we can quantify its intensity, and then we can plot those on a log-log curve here. And the slope should be 2 if we really understand that the dye is in the outermost layer of these particles. And you know, the membrane has a bit of a thickness, like 5 to 8 nanometers, and the particles around 80 nanometers. So you might expect some alternation, but this data really does lie along the line of R squared, which was pleasing. And you can see that the sample is quite homogeneous. So the cluster of points is at the center. And then we varied the pH, and this is relevant because when these particles are prepared, same thing for any of the genetic medicines or vaccine particles, they're often made at pH 4 and lowered to pH 7. And of course, in endosomal release, the pH drops too. So understanding the difference in particles at different pHs is important. So when we check out the same sample at lower pH, we do notice that this slope increases, and that is consistent with the understanding that the particle structure at lower pH reassembles a bit to have at least a couple layers on the outside. So this dye is now, you know, it's not filling the particle. If you went all the way, that'd be R cubed, but it's not just on the surface. So we get a slope between 2 and 3, and you can see from the cryo-EM pictures from the Cullis group that there is a structural difference. They look very thin on the right and a bit thicker on the left. And then we wanted to compare this data. This is a first look with this technique at different lipid particles. This is an example of the deoxyribosine formulation with no ionizable lipids, where we also expect a few layers. And again, we got a slope between 2 and 3. So it's an interesting technique for looking at what we want to do now is sort of screen different recipes where we have associated cryo-EM structures and look at the loading as a function of drug and particle recipe to really understand the biophysics of their structure and how these things might affect endosomal release, following them into cells. So here's another example. Now this is a loaded particle as opposed to an empty one, which I showed before. This is the Onpattro particle loaded with siRNA, which are loaded at labeled at a low fraction. And this is different. It's on the same scale as before. You can see that all of the data is in the center rather than lying along the line. If I held this up to the other graph, there'd be much more points along the R squared line. We took a lot of this data because we were feeling a bit insecure about it at the time. We expected it to spread out, but then we finally shared it with the Cullis group, and they had also looked in cryo-EM. And this is really consistent with a different kind of structure, which is consistent with the siRNA kind of bundling up into little blobs or aggregates inside the particle. So when this data is at the center or uncorrelated, it means that the brightness or the amount of siRNA per particle isn't scaling as the size of the particle. And it seems to be because it's loaded in these sort of blob-like ways. So our data helps confirm that picture. And these are some recent cryo-EMs of that where they look kind of blobby. So this was an unexpected result, but it shows how a detailed look at the size and intensity of the particles can give information on the structure. And so other applications of this are we're starting to look at particles with ASOs loaded on the surface or other kinds of medicines. And we feel like this technique can give us a handle on size loading and the scaling law. So that gives an idea of the structure. And this is all done in solution with nothing tethered down, and the ability to dynamically change the pH and then eventually follow the particles into cells in a microfluidic platform, which is different from separately freezing them out. So we're excited about that. Okay. And then, yeah, this slide again illustrates the ability to count dyes per particle to calculate the loading. And so we validated this technique on siRNA loaded particles with 1, 3, and 10 dyes, made sure, for example, that other effects weren't interfering with our ability to make those robust measurements, which excitingly is working out when looking over a large number of particles. So just to summarize the method, when we are interested in size and diffusion, we make measurements of very long trajectories at low laser power. When we want to count, we'll photo bleach the drugs at a high laser power. And then that gives us the simultaneous size and loading distribution calibrated precisely by the photo bleaching data per particle. And then we've moved a little bit on to next steps. So one really nice thing about this platform is we're really curious about dynamics. So once we can take a look at, the size and the loading of the particles and their structure, we're curious, how does that dynamically change when they enter the cells and also when they're released into the endosome? So we're looking at these right now in our glass cells, but we've started to vary the pH and look at some of the dynamics. So some of the questions we had are, how quickly did they fuse? And how do they respond to this lowering of pH? Can we look at fusion and release artificially, control variables, and then relate that to artificially designed endosome or artificial cell, and develop eventually an off-chip platform for looking at delivery? Those are some of the big applied questions. And just following the particles into the cells itself is very exciting. So this is a baby step where we confine the particles loaded with drugs into the CLiC device, and or just loaded with dye, we change the pH from higher to lower and just watch. And what we did as a first step is we loaded them with red and green dyes, and we're looking for FRET. So FRET occurs when the dyes are very proximal. You can shine a green laser and get red light out from transference between the proximal dyes. And this is an assay we could then run, inside a model endosome or inside a cell, load red and green particles, and then see what happens versus time to them. Here, we're looking at them fuse just to each other as a proxy for further steps. So we can take movies of the diffusing red and green particles. So here's a well that happens to have a few at short time, and then at later time, we can see that they have fused. This is a bigger blob, and we're shining green light and getting red light out. And then this is preliminary data, so this is not in the paper I referred to before, but we see the intensity in both channels, and we can track the fusion and give it a time scale and so on as a function of recipe of the particle. And then this is a movie. So you have initially some particles loaded in the well, and then this displays FRET. So as a function of time, we definitely see fusion in solution. So there was a question of whether it might only occur in the surface or non-solution, but it's occurring in solution. And then as time goes on, the FRET increases. So this is displayed in one channel where green is initial intensity and red is FRET. And then we can track that versus time. So the kinds of questions we have are, how do modifications of the recipe and structure of the particle influence those dynamics? How does endosomal release actually connect at the microscopic level to therapeutic potency? And so coming back to the start of my talk, we'd like to develop a through line of understanding through these scales. And we're working with a number of people on this at the Michael Smith Labs and some of the industry scientists in Vancouver as well. So this is on the one hand, very fundamental biophysics about the structure function of these new kinds of particles with complex polymers. And on the other hand, very applied in that it can help develop new genetic medicines and vaccines. probably coming to the end of my time. I don't have a clock on this screen, sorry. Yeah, and just so to summarize, we're very excited to be doing this work. We hope that our platform gives a unique insight by having, untethered free single molecules, particles, and cells in controlled conditions. And we're very happy to collaborate with others. We've just moved into a new lab that's been built up around us during the pandemic, so it's been a pretty intense year. And I hope I've spoken well at 6:30 in the morning. So it's the year for challenges. And we're just excited group of single molecule biophysicists hoping to make an impact, and also happy to connect with folks at AstraZeneca and England, Sweden, and so on. Once travel opens up again, please come visit. It's a really lovely place in Vancouver. All right, and then with that, I'll move to my final slide with my team. And so as I mentioned, this work has contributions from a team of biophysicists. In particular, postdoc Albert Kamanzi took most of the data with a grad student Yifei Gu, and strong support from a research associate Romain Berti on the analysis. Bunch of this data was taken during the pandemic before we moved, so that was very exciting. And collaboration with Peter Cullis and his team on the samples, Frederick Hook on helping us solve the surface sticking problem in his team in Sweden, and Mark Sutton is a very strong analysis physicist at McGill, who helped us develop a lot of our analysis tools. So really a team effort, and then scientists at Precision Nanosystems and others who have strongly guided the impact and importance of the work, which we appreciate. So thank you.
Great. Thank you very much, Sabrina. I have a very quick question. I looked at your earlier work and I understood that you were using the convex lens to actually generate the film of liquid that you were confining. And so you had a variable thickness that was reflecting the convex nature of the lens. But as I understand it, you're now using effectively some sort of a lid system to make these wells that you're confining the molecules in. Is that the case?
Yes.
Okay. So then when
there's a whole bunch of questions. So the very early work, which was in my postdoc, I started with a lens on the liquid as a way to look at single molecules and particles one at a time. But then it was very clear that that device is subject to a lot of problems like evaporation and so on. So instead, if you use the lens to bend the roof of a flow cell, we can also do it. There's other ways to bend glass too. And then the liquid is between two thin sheets of glass. So it is still the case in some studies that you can use interferometry, and you could look, here in one thin sheet of liquid, film this thick, and then look over here and it's thicker. But in other studies like this one, we find it convenient to use one height. So we can put posts, so we can seal the glass on features and then have one height. So it's
okay, I
as
see.
you wish.
I'm obviously focusing on the technical details, and both David and I were thinking along the same lines and wondering how you address the issue of preventing surface absorption to the glass surfaces. So do you have, it sounds like what you mentioned in your closing slide, that might need another talk to go over.
So it is tricky, and that actually took us. That was the hardest part of our project. In the literature, there are, of course, many ways to do this, and we tried a lot of them and they kind of failed. But in the end, some methods did work. So Albert, the postdoc, probably spent the most time trialing surfaces. But But yeah, the issue is that the glass is negatively charged and the particles are supposed to be neutral, but locally they're not. And so unless you have really perfect coverage of the surface with a good charge for the species at hand, they'll stick. So we have developed that, and we have an understanding of that, and we also work with some surface chemists on the last step of that, and that's been very helpful. But yeah, you need to protect the surface, and it's you know, complex multi-step polymer reaction as usual, but it works, and in the end that's fine.
Yeah.
Once you know what to do.
Well,
yeah.
we'll check that out in your paper. So we have a couple more questions.
Yes, we do. So Tim Craig says, this is a lovely talk, and how difficult is to fabricate, how expensive are the flow cells?
Well, so we have a micro nano fabrication facility, and we do the fabrication. I guess the real answer to that is, if you were gonna fabricate them yourself, you have to get a mask, which is like a few $100s, and you have to have people trained and do all this. But our startup company also does sell these flow cells, because they make a bunch of them. I think they're not very expensive. We reuse them. So yeah. So if, for example, like in these experiments, my postdoc would use them one day, and then he would dissolve the tape and use them again. So the effective price probably comes out as, I don't know how many times he uses them before he breaks them. But it probably ends up being a few dollars or something, because there's the cost to make them or buy them, but then you use them many times, so their effective cost is quite low. But yeah, so it's not a limiting factor at all. It's probably a lot cheaper than most biological reagents once you factor in all the multiple uses. Yeah, you just have to not break the glass, but they don't tend to. The new students might, but the experienced ones don't. They use them 100 times or something.
Okay, and Olivier Doss says that this is a very cool technique and work, and is asking if in your single molecule FRET traces, the FRET efficiency does not change within one frame as expected, but changes gradually for a single molecule binding event between donor acceptor. Why is that?
So for the graphs that I showed of FRET, like at the end, just our kind of preliminary data, next steps, I think that's what I showed as a graph. That must be what he's referring to. Those were particles filled with many molecules or dyes, and we were looking at the fusion. So, and it was quite slow. So the reason it looked smooth is because there were many molecules with dyes in the fusion of those, you know, mesoscopic particles. We do take data with single dyes, like a single antisense on an RNA or something like that. And then that's very discrete when we track red and green, and there's an event that happens immediately. But what I showed there was large numbers, many molecules in a particle and a slow event. So that was, that's why the curve looked very slow and smooth, large numbers.
Great, thank you. Okay, well, thanks for getting up so early, and thanks for such an interesting talk, and also to the other speakers in this session.